So I'm very pleased to be speaking to Sarah Stewart, who got in touch with me after some Facebook posts about the Algologies project and told me about her background in phycology or algology as we're calling it. But she's got a very diverse background as well in other areas to do with taxonomy, and I'll let her introduce herself. Thank you, Sarah.
Okay. Thank you, Anna. Hello, my name is Sarah Stewart, and I am currently a PhD student at the Oxford Internet Institute. I'm studying how open data and digital collections are influencing and changing the research practices of scientists, particularly those scientists who are studying biodiversity. I'm also a data librarian, but before that, I used to study seaweeds and freshwater red algae. So that's me.
And some of the project that I'm working on is to do with the exploration of the seaweed, but also to do with the relationship of women in science to seaweed collecting and how that's evolved into, you know, wider roles for women in science. So I'd be really interested to hear more of your thoughts on that. And also I'm thinking also a later question will be about your expertise in the data and how that's changing science. How is that impacting phycology and the study of seaweed? Because we have these vast seaweed collections, not least created by Mary Philadelphia Merrifield, in whose footsteps I am stepping in Brighton here. So I'd love to hear also about that. But yeah, tell me everything.
Hey, well, phycology is actually a really great science, which has actually involved women very much from the very beginning. I mean, it was considered sort of, as you mentioned in our discussion earlier, it was considered to be sort of an acceptable form of botany for young women to, you know, to look after in the Victorian ages. Often young middle-class women would be encouraged to study nature and to look at it. But on the other hand, botany was a bit of a contentious subject because of the Linnaeus system of taxonomy, which was very sexual. And so that was kind of considered something a bit taboo, you know, for young, decent young women to study. So seaweeds, I think, were less well known. In fact, the group of seaweeds, mosses, ferns, they were known as cryptogams, so effectively secret or hidden sexuality. So in that sense, because the life cycles weren't very well understood at that time, you know, and they weren't considered to be very sexual, you know, as opposed to, say, a, you know, Linnaean system, it was considered more acceptable for young women to study seaweeds than other types of things. There's actually a beautiful pressed specimen in the Natural History Museum. It's a small book, a small fold-out book that's been digitized. I'll have to send you a link to it, but it basically is from the early 1800s, and it sort of shows all the different seaweeds collected along the British coastline. All, you know, red algae, brown algae, green algae, which are the three major groups of seaweeds that you'll find, and it's fascinating just to think that somebody made this. So yeah, I mean, it was very much sort of an amateur science, but that amateur science, like a lot of taxonomy, evolved in, you know, from this very descriptive natural history into a much more rigorous, or I guess experimental scientific type of pursuit. And actually, again, women led the field in that area. I'm thinking of Kathleen Drew Barker, who was actually revered in Japan because she was one of the first women scientists studying the life cycle of red algae, which is now being used, you know, for culinary purposes in Japan as well. So that's amazing. I mean, if you go to, I believe it's in Kyoto, but there's a statue of her that you can visit as well. So that's sort of one area. There's also a dinoflagellate. So again, algae is sort of a weird group because they're, you know, it goes from everything from tiny microscopic single cellular protists all the way up to giant, you know, the giant, you know, brown kelps that you'll see in the kelp forests in California.
We have kelp forest off the coast of Brighton, and we've had quite interesting things with they're trying to renew this kelp forest, and I think there's been activists as well putting things down in the water to stop the fishermen being able to trawl at those levels to damage the kelp. So there's been a bit of kind of a bit of a contested area there, and a lot of sort of nature activism to do with the kelp forest off Brighton.
There is, and that's really great. It's really great to hear that that's being protected, because those marine habitats are so important. I mean, just having the kelp there and conserving those kelp forests will actually help increase biodiversity for many other groups as well, because algae are so fundamental for marine life. Even the single cellular phytoplankton, which are in the sea, like the coccolithophorids, which is another group within the algae. Algae is sort of a strange taxonomic, it's like a really mixed bag of things. So when you mentioned earlier that idea that algae is kind of between plant and animal, it in some ways I can kind of see that, because it is very much a group unto itself. I mean, it evolved, the different groups of seaweeds supposedly evolved by capturing bacteria and sort of harnessing them. So you have the green algae, which is sort of taken in a chloroplast. You know, it's sort of harvested that, and then similarly the red algae took another form of bacterium to help it photosynthesize, and similarly the brown algae. So there's been this kind of taking in and sort of almost such a close symbiotic relationship between the different types of photosynthetic pigments within
I mean, even we took in these bacteria, and they became part of us as well. The mitochondria in our cells, the energy kind of houses in the cell, the chloroplasts in the plant kingdom, and now the seaweed. I mean, at the heart of all of us, we are these bacteria. And it's really fascinating to think of it
yes
like that as well.
no,
And the importance
really,
of the kelp forests and things like that, because I was fascinated to hear that, you know, that the sea and the seaweed are such an important thing for protecting the climate, and they're absorbing so much carbon, and actually that seaweed farming for things like agar or for food is actually carbon negative, isn't it? It's really beneficial for the planet. The more seaweed we can actually encourage is a very good thing for preventing climate or mitigating climate change.
It really is. I mean, even the coccolithophorids, which I mentioned earlier, they're sort of a microscopic plankton, but they have these plates that are made of calcium carbonate. So again, right there, you have a very active way of drawing in carbon and sequestering it in these things. And that's actually what makes chalk. So if you think about the Brighton coastline, all of that is fossilized. Well, other kinds of things as well, but mostly coccolithophorids within that chalk. So that's quite.
Is like that sort of crusty stuff you get on this kind of seaweed?
That might be. It's hard to see, but does it look like honeycomb? It's like, I'm just trying to peer very closely at it.
Yeah, very tiny.
Yeah, that's probably a bryozoan. So that's actually an animal. It's related.
It's living on
it's related to sponges, but they form these sort of crusts of cellular structures on different things.
It's almost like a lizard skin when I look at it.
Yes, yep, that's it. That's definitely a bryozoan. And it's quite funny because those are often mistaken for seaweed. Sometimes they'll form these blade-like structures, and you'll find them washed up on the shore. And
but I mean, this is a seaweed that is on, isn't it?
That's definitely a seaweed. It's just covered with
seaweed, but they're just all over it. Yeah,
but you'll probably find some. I mean, they're very. I haven't been to Brighton in a while, but
really encrusted with sand or something. I wondered if this was the calcium carbonate.
That looks like another bryozoan, or
a really full on one.
Maybe, yeah, but yeah, they can get really full on. Like, they don't even have to cover seaweed. Sometimes they just grow in these blades, and they almost mimic seaweed. Like, they create these sort of really interesting, really sculptural, really interesting three-dimensional structures. It's an animal, yes. Yeah, it kind of covers things. It's a bit like a coral. And actually that's interesting too, because there's the coralline red algae, which you will probably find around Brighton as well, that they sometimes will form crusts on rocks. So it's like a reddish colored.
That I've seen, where you've got this kind of very like threads, as if it's somebody's been sewing and they've thrown the red threads down over a rock. like almost as if it's been felted or wrapped in thread.
That might be a different species, but this one actually looks like a rock or a lichen. Like it's very hard. It
Ah,
actually, again, it has this calcium carbonate type um, structure to it. So you actually have some that look like coral, like miniature tiny coral, and they're hard, and they'll become very brittle and break when they die. Or you get these sort of crusts on rocks,
mm.
which are, they have a sort of a purplish red color. If you see them, you'll notice them, I think,
I
because they're
mean,
very.
I've seen rocks that are covered in this thread-like stuff. There's a lot of
Mm,
that. It was almost at one point, sometime last year, when we walked down to a part of the beach that had the tide was out, because normally that bit the tide's really in, and there are steps down, but they're sort of semi-washed away and they're really slippery. So it's like a bit of beach people don't tend to go to, but we decided to go down there and explore. And the whole thing was like a Martian landscape. It was covered in this fine thread-like red weed, the whole kind of
mm,
area. Yeah, it was like Mars or something. And I think that was the moment when I started to notice seaweed. It was like, this is so strange. So I've been trying to go to different parts of the beach, because this sort of bag full of things that I collected yesterday is a very different bag than the types of things I was collecting on other days, and things from different parts of the beach. So it changes quite substantially. Yeah. It changes.
yeah, yeah. I mean, seaweed are amazing. Like I'm surprised more people don't. Well, algae is amazing. Just the whole
Mm.
group is really amazing. I
I
mean,
mean, we just ignore it, don't we? But that's what's so strange.
yeah.
Like, most of the world just ignores it. And only when I was sort of forced to notice it every day changing and being so odd, and I just wanted to know more. And then the more I research, the more kind of fascinating it becomes. And I've learned about, I've read all the methodologies now of Mary Philadelphia Merrifield. I'm trying to kind of enact her advice of the times of day to go seaweed collecting and things like that. And then this interest in the relationship of agar jelly that you mentioned, that is used now in every lab in the world, and was discovered by Fanny Hess in Robert Koch's lab, and to be used in science. And recently I stumbled across a report about the use of British seaweeds in the production of agar, a wartime report. So I want to kind of explore that, and trying to do more research and get actually hold of that report to study it further, and see what the results were, because it was a wartime report. They were worried that the seaweed supplies from Japan weren't going to come through, and because of all the research in penicillin, they needed as much agar to do the microbiological experiments as possible. So so there was this move to try and find the British algae for agar, which is kind of fascinating. And then that led me to have some other strange questions, and maybe something, or maybe it hasn't happened, or like with these seaweeds I was reading. So these ones that I've been collecting, that might be good for agar, and then when I was doing my experiments, they were to do natural dyeing with seaweed, they were turning to jelly. So that led me to kind of think about these things. But I was also wondering, is it possible, like I work a lot with CRISPR technology and synthetic biology. Is anybody modifying seaweed? Is that a thing? How does that work? Is there synthetic biology? Are there moves to make? Like, could we have a genetically modified British seaweed that made a lot of agar or something like that? Um, or
I
can
think
we
it's on the
partner these?
it's probably on the way, but I think it's taking a bit. Like, it will probably take a bit of time to get there. So I think, from what I remember, is there one seaweed that has had its full genome sequenced at the moment? So once that's happened and a bit more experiments are being conducted, I can definitely foresee that there will be a lot of um, the potential for modifying seaweed could be
Hmm.
there, although I think it's still sort of in development at the moment.
There's only one seaweed that's been whole genome sequenced so far.
I believe,
Yeah.
I'll double
Well,
check. I'll
it's
double
it's
check
not
on
a
that, but
huge area at
it's
the moment.
Not that I foresee, although there's a small brown seaweed. It's another native of Brighton, so it could be
Mm.
could be British, but Ectocarpus siliculosus is sort of being developed as a model seaweed, and
Can you
that's
spell that? Ectocarpus.
uh, ECTO, like
Yep.
ectoplasm, CARPUS,
Yeah.
and then it's siliculosis, SILIC,
Okay. I think I've got it.
well,
Siliculus.
siliculosis.
Yeah. Great.
It's a tiny. It actually looks a little bit like laundry lint. It's a small brown, effectively a relative of kelp, so it's within the brown seaweeds.
Mhm.
And they're modifying that as a model organism.
Oh, I'll have to look into that. That's a good tip. That'll be another part of the research that I'll have to look into. Because do you know if they have very big genomes, seaweeds, like plants,
I
or
expect it will vary quite a bit because, again, depending on what you're sequencing, anything from, a protist, which, is that a plant? Is that an animal? What is that? Because euglenoids are
Mm,
considered to be part of algae, as are dinoflagellates and coccolithophorids as well. But then you've got really massive kelp, and they can have all kinds of different things. And also the life cycle of seaweeds is very complex as well. I mean, again, going back to our earlier part of the interview about women in science, there's a dinoflagellate called Physteria, which is actually toxic. It produces the red tides, and that was sort of the life cycle actually has 7 different stages, and that was sort of unraveled
do we have
by
that in, do we have that in the UK? These red
I
tides.
don't think so. No, no. I'm just trying to
There's me
remember.
going out collecting loads of red seaweed. I don't want to be
Yeah.
collecting.
Oh no, it's you won't. It's microscopic. It's like a
Okay.
tiny little thing. It's just dangerous when it gets to huge quantities. So
Uh,
then you have these red tides. It's often on the Gulf Coast of the southern U.S. like Florida, Louisiana, and it's just really toxic.
so it's a bit
But
like
that's not.
when we talk about algal blooms. It's that sort of thing, is it? Yeah. Yeah.
Yes, very much so. And
We
actually,
do
seaweed.
sometimes get algal blooms in Brighton, just near here, outside, yeah,
Mhm.
where I'm sitting now. Sometimes we see algal blooms coming down in after the rain and the hot weather and things. Sometimes we get it.
Yeah, and this is similarly with the phosphorescence as well. Like some of the sea, actually chondrus, the seaweed that you held up, if you see it in life, when it becomes reproductive, so at a certain part of its life cycle, it'll actually have a blue metallic sheen to it, and it will look really iridescent underwater.
Mm.
And that's just sort of an interesting thing about how seaweed can change throughout the season. So it will sort of, similar to a plant, it will sort of have a reproductive cycle where it's sort of blooming, although it's not, it's releasing its gametes,
They
and then it
really
will.
do change, and after a year of kind of being there every day, it's just incredible. Even the tides, like the different time of the tide, it looks. I mean, we went out at kind of just past low tide, so it had been really drying out for a long time, and it was like quite a barren landscape, just these things kind of remaining really, and then the rest of it all washed up like this stuff which I've been kind of collecting. I've made it my thing just to pick up the ones that are loose because of the fact that they're so good for carbon capture and things like that. I don't want to be going, and I don't advocate picking it, but like finding this loose stuff and like exploring it is really fascinating to do. Mm. And
Yeah.
you talked about taxonomy as well, didn't you? And
Mhm. Yes. So, looking at women in how women have developed a lot, because women have actually led a lot of the taxonomic investigations into seaweeds. So I'm thinking, in our current time at the Natural History Museum, you have experts like Dr. Juliette Brody, who her specialty is coralline red algae, but she's another red algal phycologist. She might be a really good person to interview, actually.
mm,
So I'd recommend getting in touch with her. Um, Many of the, the development of, sorry, I can't speak anymore. The development of algal taxonomic groups has actually been led by women, working in the field. So they'll go out and collect specimens. I mean, currently with the taxonomy, much of it is, it's molecularly based, but also using the morphological characters. So what seaweed actually looks like, and also its life cycle and its ecological stages as well. I mean, you mentioned that at low tide and it was very dry and it was only that brown fucus that was left. Looking at how seaweeds stratifies themselves on the beach is a really good clue to their ecology as well. So fucus and sort of brown algal kelps like fucus are really drought resistant. So often you'll find them quite high up on the tide line, and they'll sort of desiccate, but then they'll come back when the tide comes in again. So it's quite interesting just to sort of have a look on the shore and see how the seaweed sort of stratifies itself. And you'll often find a lot the red algae, because they're very good at photosynthesis at very long wavelengths of light, so they tend to be more in deeper waters. You won't find them so high up on the shore.
Yeah, so it'd be hard to see them in real life growing unless I don diving equipment, which is unlikely, or use an underwater drone.
Yeah, or you can see them in tide pools because they don't like drying out, but you'll often find them
we've got loads of rock pools just here. This is part of the thing, and it goes out a really long way. It goes out, and you can see the tracks of Magnus Volk's Daddy Longlegs train, which cuts through, which he built this sea-going train. Actually went through the water, but apparently at high tide it really struggled to get along. It only lasted 5 years, but you can still see, and he was this sort of pioneering inventor that built a lot of things. In fact, he built an electric car as well in the sort of 19th century. So he was a crazy dude. As one of my friends that's working with studying the Volks's Daddy Longlegs says, what could go wrong? Electricity and seawater. Because it was an train. So yeah, you can see the tracks of that, and then you can see the shores go out, the rock pools go out even further, so you could really get out there. I've decided that I need to get further out, so the more I do this, the further out I'm going to do. I'm always scared of slipping over. I was talking about, I've been writing about this for the blog. It's like, as I was a child, I was always get so far out, and I'd be like, I can't get back in, and my mother would have to come and carry me in, because I'd be like, I've scratched my knee on all the barnacles and stuff. It's not without risk, this artistic project.
No, it's not.
The Society for Applied Microbiology article talking about all the work to look for the seaweeds to make agar talked about how they were surprised there weren't more accidents in the project. So it's quite interesting to think of all that. And so with seaweed now, so we're at the sort of the early stages of seaweed genomics, it seems. So that's obviously going to change a huge amount. I'm thinking about how the sort of Archaebacteria kingdom has now split into two kingdoms in sort of two domains of life now, and archaea and bacteria, because they realise that although they do look the same, they appear the same, actually genetically they're as different as we are from elephants or fish or some, they're huge, maybe more different, more genetic diversity. So there could be massive genetic diversity in seaweed, completely untapped, and thinking of them as a carbon negative kind of crop and things like that, the benefits for climate change. It seems that we're at the start of a new era, and where your new interest of data and big data, and how big data changes science, and this sharing of it, that could turn out to be something really dramatic for phycology or algology, as we're called.
Yes, yeah, I think it will be. I mean, I'm still in the early stages of my research at the moment, but what the patterns that I'm seeing is that scientists are actually now starting to explore these collections that are being held in natural history museums. And this is sort of considered in a way sort of a locked up set of data, because you have these collections, you have a physical specimen, but that might relate to several genetic sequence or other molecular data that's been recorded. It might relate to field notebooks, it might relate to ecological data. So it's just how can all this data be linked up
Mm,
and used in a way that's effective. So no, I certainly foresee that there will be big changes, particularly once a lot of the genomes for seaweeds start being sequenced. I think that will really open up a lot of interesting
mm,
um, questions about the current accepted taxonomy
mm,
versus what it might actually be. I mean, it could be very interesting. I mean, particularly because the lineages that have produced seaweed are so ancient. I mean, they go back to these early archaeobacteria lineages as well. So I think there'll be some really interesting
mm,
findings coming from this. So I'm really curious to see how scientists will use data to kind of tap into the collections and find
mm.
ways of linking data and finding new patterns that will produce new taxonomies.
So we're kind of at a stage where we're right on the verge of a whole kind of um, new sort of spirit of algology or phycology for the world, where scientists are going to be as obsessed as they were during the height of the algology hobby time, and it was as popular then.
Mhm.
So, and especially with its particular impact that it might have on the climate and things like that, and
Oh, yeah, yeah, definitely.
the
Mm.
potential for synthetic biology research. I think it's almost like it's an untapped area. I certainly haven't come across any papers around this kind of area. I mean, I'm in the early stages of this research, and I'm grateful to receive anything from anyone who's kind of listening to our discussion and things like that. So I'm aware of that, but it seems like it's a really exciting area for now. Thank you so much, Sarah, for
Oh,
talking
you're welcome.
to me about the work. It's been wonderful.